So, when Charles Darwin published On the Origin of Species, he totally changed the game. He gave us this revolutionary idea, evolution by natural selection. But, even he admitted there was this huge piece of the puzzle missing. He knew selection worked, but he had no idea how traits were actually passed from parent to offspring, or where new traits even came from in the first place. This is the story of how genetics finally filled in those gaps, completing one of the absolute greatest ideas in all of science.
All right, so how did we get from Darwin's big puzzle to the complete picture we have today? Well, our journey is going to start with the exact questions Darwin left behind. Then we'll follow the early clues that scientists started to uncover, witness the major genetic breakthroughs that changed everything, and then see how all these ideas came together in what we call the modern synthesis. It's going to bring us right up to how we understand evolution today. Okay, let's dive right in. To really appreciate the solution, we've got to first understand the massive problem that scientists were grappling with back in the late 19th century.
Darwin's theory was an absolute masterpiece for sure, but was it a finished work? It's really tempting to think so, but the scientific community knew there were some serious, nagging questions that just had to be answered before the theory could truly stand on its own 2 feet.
And here they are, the 2 big ones. First, the mechanism of heredity. The popular idea back then was blending inheritance. Just think of it like mixing black and white paint to get gray. Now, if that were true, any rare, new, advantageous trait would just get diluted out of the population super quickly. It would disappear into the average. That would make natural selection completely impossible. And second, where does new variation even come from? For natural selection to work, it needs options to choose from. Darwin, he just didn't have a good answer for either of these. So, with these huge questions just hanging in the air, a whole new generation of scientists started searching for clues. Let's take a look at some of the early investigations that began to point everyone in the right direction.
First up, we've got Ernst Haeckel with his really famous, if a little controversial idea, ontogeny recapitulates phylogeny. Whoa, what does that mean? Well, in simpler terms, he believed that the development of an individual organism, that's its ontogeny, basically repeats the evolutionary history of its entire species, its phylogeny. For example, he pointed out that the embryos of fish, birds, and even us humans all have these things called pharyngeal arches, which look a lot like gill slits. It suggested a shared, ancient aquatic ancestor. Now, we know it's a huge oversimplification, but it was a really powerful idea linking how we develop to how we evolve.
Next up is August Weismann, and he made a truly critical distinction. He proposed that an organism has 2 totally separate types of cells. You've got somatic cells, which make up your body, your skin, your muscles, everything, and then you have germ cells, which are the sperm and the egg. This was a radical idea. It meant that only the germ cells pass on hereditary information. So, changes to your body, like building muscle at the gym or getting a scar, could not be passed on to your kids. This directly challenged some of the oldest theories about inheritance.
And to test this theory, Weismann did this classic, kind of gruesome experiment. He took a bunch of mice and cut off their tails. Then he let them breed, and he cut off the tails of their babies, and their babies' babies, and so on for 22 straight generations. Now, if traits you acquire could be inherited, you'd totally expect that eventually baby mice would be born with shorter tails, or maybe even no tails at all.
But that is not what happened. Not even close. Every single mouse in every single one of those generations was born with a perfectly normal long tail. The result was just undeniable. Weismann's experiment was a death blow to that older idea of Lamarckism, the inheritance of acquired characteristics. This completely cleared the intellectual deck and paved the way for modern genetics.
So with the old, incorrect ideas finally disproven, the stage was set for the real mechanism of inheritance to be revealed. And that brings us to the monumental breakthroughs that answered Darwin's first big question.
And here he is, Gregor Mendel. Working quietly in his monastery garden, Mendel's experiments with pea plants showed that traits weren't blended at all. Instead, they were passed down as these discrete, individual units, what we now, of course, call genes. His laws of segregation and independent assortment explained exactly how these units are shuffled and passed on, preserving variation from one generation to the next. This was it. This was the elegant mechanism of inheritance that Darwin's theory so desperately needed.
But that still left Darwin's second question unanswered. Where does the new variation come from? Well, Hugo de Vries came along and provided the answer with his mutation theory. He proposed that new traits don't just appear gradually over time, they pop up from sudden, random changes in genes, which he called mutations.
These mutations are the ultimate raw material, the creative engine that provides all the novelty for natural selection to work on.
Okay, so now we have the rules of inheritance from Mendel and the source of new traits from de Vries. So the next logical step was to take these rules and apply them not just to a single family of pea plants, but to see how they change entire populations over time. And this is where the Hardy-Weinberg principle comes in. The best way to think about it is like a scientific control group or a null model for evolution. It mathematically describes this perfect, hypothetical, idealized population that is not evolving at all. By setting this perfect static baseline, it gives us a way to measure real-world change against it. So, for a population to be in this non-evolving state, 5 super strict conditions have to be met. No new mutations, totally random mating, absolutely no natural selection, a very, very large population size, and no gene flow, which just means nobody is moving in or out. Now, in the real world, these conditions are almost never all met. And that's the point. When any one of them is broken, the population's gene frequencies will start to change. In other words, it evolves. And this is how we can actually measure it. Using these really elegant equations, scientists can assign variables to the frequency of different versions of a gene, or alleles. We use P for one and Q for the other. By plugging these into the model, we can calculate the genetic makeup we should see if the population isn't evolving. When the real-world numbers don't match that prediction, we know evolution is at work, and we can even measure how strong it is. So now finally, all the pieces were on the table. Darwin's natural selection, Mendel's heredity, de Vries's mutations, and the powerful math of population genetics. It was time for the grand unification. And this is what we call the modern synthesis. You can almost picture Darwin's theory of evolution on one side of a great canyon, and the new science of genetics on the other. For decades, they were pretty much separate. The modern synthesis was the bridge that finally connected them, creating a single, incredibly robust and comprehensive framework for understanding all of life. And this incredible unification gave us our modern, super precise definition of evolution. It's not just some vague idea about change over millions of years. It is specifically and measurably a change in the frequencies of alleles within a population's gene pool from one generation to the next. Simple as that. And, this wasn't the work of just one person. It was a true team effort of brilliant scientists. You had Theodosius Stevjansky, who integrated genetics into studies of real animal populations. Ernst Mayr, who developed our modern concept of what a species actually is. Fisher, Haldane, and Wright were the mathematical geniuses who built the foundations of population genetics. And it was Julian Huxley who gave the whole movement its unifying name, the modern synthesis. So where does that leave us today? Let's take a quick look at the core processes of evolution as we understand them now, standing on the shoulders of all those giants. Today, we recognize these 5 fundamental processes. It's actually pretty simple. The first 3, gene mutation, chromosomal changes, and genetic recombination, are the engines that constantly create new genetic variability. They're providing the raw material. Then, the last 2, natural selection and reproductive isolation, are the processes that shape and guide that variation, leading to adaptation and eventually the formation of brand new species. So ultimately, the key takeaway here is this. Modern evolutionary theory is not a replacement for Darwin. Not at all. It's the completion of his vision. It stands firmly on 2 massive pillars: Darwin's profound insights into natural selection and the powerful predictive science of genetics that finally explain the mechanics behind it all. And this really leaves us with a final thought to chew on. We've built a bridge, we've answered Darwin's great questions about inheritance and variation. So, what's next? What are the new frontiers? What are the great unanswered questions that will drive the next synthesis in our understanding of life? Now that is something to think about.